Okay, so thank you very much for the introduction, and thank you for the chance to present some of the work we have been doing lately. So I will not be talking about exclusively the work we do at Calixar, but I will really rather be reporting on the recent outcome of a collaborative effort that we have together with the THERMO FISHER SCIENTIFIC. So, of course, it deals with the expression or difficult to express membrane proteins, since they are really extremely important for drug discovery. I will focus on one case study to actually tell you a little bit more what kind of take-home message we can get out of that. So the focus of the presentation is basically a little bit described here. So we'll be talking about strategies to optimize expression levels of membrane proteins using the XP293 and XP-CHO expression systems. Well, we'll also tell you a little bit about the new developed XP293 that our colleagues at Thermo Fisher are developing, and this have something to do with inducible NGNTI minus engineered cell line. There's something wrong with the microphone. So I will tell you also how we are using Calixar patented technology to evaluate the quality and the quantity of the challenging membrane proteins we express. Something which is extremely important is not only to express or to overexpress, it's to overexpress healthy protein. What we want at the end is not to have tons of aggregated protein, but it's have milligrams of high quality proteins. And this is why the collaboration between Calixar and Thermo Fisher is actually nice, because we are combining expertise. Sorry. Okay, so the focus of the presentation will all be, like I said, we'll be focusing on KCC2, which is one of the key proteins in drug discovery, and this is why we are collaborating on that. So KCC2 is a neuron-specific potassium chloride cotransporter that controls neuronal chloride homeostasis, and it's absolutely required for involved in different neurological disorders, including brain trauma, epilepsy, autism, and schizophrenia. KCC2 is absolutely need to be glycosylated and also phosphorylated to be active, and we have been describing basically this topological structure with different glycans and different phosphorylation that we could actually identify in this paper that I will cite very often in this presentation. So it's really a nice target for this assessment, because it needs to be glycosylated and need to be post-fluorescence modified, and the systems we are using, XP293 and XP-CHO cells, are actually known to be able to do that. So let's have a look to some of the data. So what we wanted to start by doing is to screen XP293 and XP-CHO cells in terms of expression of this target. So this is not a nice gel. I will show you better ones, but typically this protein migrates as in different species. One of the species is so two glycan forms and then one dimeric form that is difficult to dissociate in STS page. So we have looked into different parameters. One of the parameters is the type of vector. This one is under CMV promoter, and the other one is under EF1 alpha promoter. We looked into different DNA amount. We looked into, like I said already, two cell lines, XP293 and XP-CHO cells, two temperatures, and then we also performed work on two constructs, N-terminus tagged and C-terminus tags. The take-home message is that obviously we get better expression in these conditions, XP293, and also using the CMV promoter at 32 degrees. So this is not extremely surprising, because our colleagues at Thermo working with soluble protein get also similar tendencies. So it does not mean that you don't get expression in CHO cells, but you just get less expression for XP-CHO cells. So one thing I wanted to point out is the amount of, or the ratio of protein expressed as a plasma membrane here versus internal membrane. So we have worked previously, and we published this paper 2 years ago, where we could see that 50% of the KCC2 is actually present in the plasma membrane, and the other half of it is present in the internal membranes. So this is not surprising. We have shown this when you are using classical hex cells. Now, what is interesting is when you actually move to XP293 and you express the same protein with an N-terminus tag or a C-terminus tag, you get somehow better expression in the plasma membrane. So the ratio is somehow changing. So you get better expression in the plasma membrane in comparison to internal membranes. So this was true for N-terminus construct, C-terminus construct in XP293, but it's also the same thing when you work with XP CHO cells. So you have pretty much the same tendencies. So so better ratio of KCC2 plasma versus internal membranes for XP293 and XP CHO in comparison to classical ancestor, which is HEC293. So, and this was what we published previously. So we therefore focus on the N-terminal tag because we have shown in the past, in collaboration with AstraZeneca, that when the protein is tagged on the C-terminus, it's actually not functional. So something is wrong when you add even a short tag on the C-terminus. So therefore, we focus only on N-terminus tag to be able to assess basically the quality of what we express later on. So this is where we are using a Calixar approach, which is basically a patented technology using non-denaturated process to avoid different aspects like refolding, mutagenesis, and still have an impact on the stability of the protein and maintain functional and structural integrities. So this is, I don't have time to go into detail, but this is using proprietary compounds and combination of those compounds to not only solubilize, but in the meantime, stabilize. I think this is one of the discussions we already had before in one of the talks by Oxford Genomic Consortium. So the compounds we come up with are, some of them are listed here, so they have different chemistry. So in this case, it's calixarene-based chemistry. So there are a couple of structures. There is a nice, well, I cannot say nice because it's one of our work. There is a review describing this in more detail, and there is a recent paper showing how we could use these compounds to actually stabilize native GPCRs and then enable fragment-based drug design. So I will not talk about this now. So, but there are also other compounds that we came up with, and they are amphiphilic compounds, not necessarily calixarene-based. Some of them is this, what we call CLIP. So they show really a good stabilization power on GPCRs and transporters. And there are other ones that have extremely different chemistry, so don't have time to go into the detail. But this is really like a toolbox to allow us to assess what do we express together with our colleagues at Thermo Fisher. So we have shown previously in this paper that internal membranes protein are extremely, or let's say KCC2 expressed in internal membranes are extremely difficult to extract from those membranes. So this is just a dot plot. In plasma membrane, you can easily extract the proteins. In internal membranes, only the positive control, which is SDS, work, and the other ones are really lousy extraction. So this was also the same result, and this was from hex cells, right? So the same result when you do Western blot. You see the soluble fraction, you have nothing, and the pellets, everything is in the pellet. Now, when you work with XP293, it seems that we are actually able to solubilize the protein, which was not the case in hex cells. So even if we start from internal membranes, it's actually able, and we were able to solubilize the protein. That was actually the first time and the first surprise. So basically, KCC2 can be easily extracted from internal membranes, which is not the case in hex cells. And this result suggests probably that KCC2 was in better folding. And then the same result was also obtained in the other cell line, which is XP CHO cells. Now, okay, it's great. You can solubilize it. You can express it and solubilize it. But are we able to purify it? So indeed, using and focus only on this construct, we could, purify it. And this is a stain-free gel. You could also, by Western blot, see that you have this double band corresponding to different glycan forms. And so we can elute specifically the protein. So typically, KCC2 can be extracted and purified from XP293 nicely. And this was from internal, but also from plasma membrane. It was from XP, but also from CHO cells. And this was, in this case, the N-terminus tag, but the C-terminus also worked the same. So it's really nice to see that this expression system allow to have quite nice purified protein. So now, how well folded is this protein? So we did size exclusion chromatography and native page, and we could see that we have monomers and dimers. And this is not surprising because that's what we actually already established in this manuscript previously published. So we could see that we could separate the dimer from the monomer by gel filtration and then evaluate that by native page. And also in the gel filtration, you could see that there is nothing in the void volume. So definitely there are no aggregates, and there are obviously two populations, dimers and monomers. And then if you look into this population in negative stain, you see dimers and monomers. You could even get some low resolution reconstructions out of this negative stain pictures. And yeah, so we could show for the first time the organization of this transporter, which is kind of highlight the function. I will not talk about this now because we don't have time. But typically, out of this XP293 cell line, we could obtain pure aggregate-free KCC2. Of course, I'm talking about human KCC2. And this was the same result starting from XP CHO cells. Now, our colleagues at THERMO FISHER Scientific, they could develop a new system to produce basically the XP293. I don't have all the detail, but you are more than welcome to talk to them. There is a poster to describe that. But typically, you could get much more expression using this new system, and this will be probably launched, I think, in a couple of months. I will have to check that with them. But typically, this system allows to obtain more than 4 to 5 times more protein with the same quality. So what we call quality is basically at least evaluate into native page what we purify. So we have pretty much the same pattern in terms of monomers, dimers presence, and no aggregates whatsoever in there. And also this was checked by gel filtration as well. So, and this is not always the case because, when you overexpress the protein, very often you also generate more aggregates. So there is always a good correlation between overexpression and aggregability. And obviously, in this case, it seems to be quite healthy protein. It gets more protein, but it also gets healthy proteins. Okay, and yeah, so this healthy protein allows us now to collect some cryo-electron microscopy data. So I show you some low-resolution maps. So I will not be showing you high-resolution maps today, but we are actually still processing this data. But definitely, thanks to the amount of protein that we could obtain, and thanks to the high quality of the protein that was expressed using XP293 system, we could actually be able to concentrate the protein to 6 milligram per ml, and then dilute some of it to prepare some cryo grids. And then there are a couple of data sets that are evaluated now. So just a couple of slides to show you also a little bit of innovation going on the glycosylation aspect. So typically, and we have heard that before, glycosylation is important for function. KCC2, when you treat it with PNGAs, you see that you have fast migrating bands, and this was when you work with hex cells. So we have shown this already in the past. So that's the reason why at THERMO FISHER they basically try to express the XP293 with the KCC2 and XP293 GNTI minus cell line. And obviously you could see that you have these double bands corresponding to class CC2, but then you have also lower band corresponding probably less complex glycan of KCC2. So now when you solubilize and purify the protein and then put that into a native page, you again get the same pattern, which is monomers and dimers, but somehow it shifted, it migrates more rapidly, which show that probably we have populations that are less complex in terms of glycosylation, which is actually extremely important for antibody discovery, and also, of course, for structural biology, because you get less heterogeneity. And then for the antibody discovery, it can be important because you get better accessibility of your epitope of interest. And in terms of induction, this is also ongoing work together with our colleagues. And so the idea is to try to tackle the toxicity of some of the membrane proteins. Some of the membrane proteins are just toxic because some ion channels, for example, start to interfere with the metabolism when you express them. So introducing this TET induction system in XP293 can maybe address that aspect of toxicity. That's the reason why they could start to induce using this TET induction system. And interestingly, you see there is a good correlation between the induction and the amount of protein that is expressed. And then if you solubilize this protein and purify it, then you also again get similar patterns. So that means it's really, we are getting more protein, but we are also getting good quality protein. So the TET induction system of XP293 is of high interest to toxic proteins I mentioned already, such as ion channels or transporters. It allows to express and purify aggregate-free protein. And recently, there is an idea of combining TET induction with the glycan complexity reduction using the GNTI minus. And this is actually also evaluated, and we are part of the evaluators for this system that also will be available. And finally, I want to conclude by telling you what's basically we could learn from this so mainly from KCC2 work, but also including other targets. What we could learn is that the BCDNA CMV promoter allow better expression than the PEF1 alpha. It seems that the XP293 show better expression than the XP CHO cells, at least for the membrane proteins we looked at. There is obviously a better ratio in terms of KCC2 plasma membrane versus internal membranes. And this is in comparison to ancestor, which is HEC cells. The KCC2 was successfully solubilized and purified using Calixar patented technology. From internal membranes, that was for the first time actually we are able to do that from internal membranes. And this was not the case for HEC cells, probably because there's a better fold. I mean, of course, this needs to be evaluated further, but that's what we have. We think it's the origin of this what we observe. The expression yield and quality enable now cryo-EM studies of KCC2, but it's going to be the same for fragment-based drug design, and then also the same for antibody discovery, pretty much, because what they have in common is the quality of the protein. So the same tendency were observed in XP CHO cells, and we also did the same work with other constructs, and we could have the same tendencies with other targets like sodium voltage channels. I don't talk about this in detail. So the modification of XP293 expression system allow to have a much better yield in terms of protein without compromising the quality. And then finally, the GNTI and the inducible system of XP293 allow to reduce glycosylation complexity and modulate KCC2 expression respectively, and this will be something to evaluate also in combination. And then finally, I would like to say that XP293 and XP CHO cells offer really a nice opportunity to overexpress difficult to express membrane protein, not only to overexpress them, but to express and allow to purify healthy protein, because that's the goal at the end. So, and finally, I would like to thank people that were involved in this work. So our colleagues at THERMO FISHER Scientific, so that are, some of them are in the audience. Please contact them directly if you want to learn more about the cell lines. And there is also a poster that describes this in more detail. Sorry, I'm going back. And then and this poster is A100, so it's the title is indicated here. And then some of my people from my team that are listed here that really were involved in this very interactive collaboration. And thank you very much for your attention, and I'm happy to take questions.